Hello and welcome to this tutorial on multiple sequence alignment using Clustal Omega and MUSCLE. In this video, we will learn how to perform multiple sequence alignment using these powerful bioinformatics tools. Multiple sequence alignment is a method used to align three or more biological sequences and identify regions of similarity that may indicate functional, structural, or evolutionary relationships. In this exercise, we will use insulin sequences from different organisms to demonstrate how to perform multiple sequence alignment and analyze the results. Insulin is a crucial hormone produced by the pancreas that regulates blood glucose levels. Insulin is synthesized as a precursor protein called pre-proinsulin, which includes a signal peptide making up a total of around 110 amino acids. Pre-proinsulin undergoes pre-processing to form proinsulin, which has about 86 amino acids. Further processing results in mature insulin consisting of two chains, the A chain with 21 amino acids and the B chain with 30 amino acids, connected by disulfide bonds, totaling 51 amino acids. Understanding the conservation of insulin's functional regions can provide insights into its evolutionary relationships and functional mechanisms. First, we will learn how to download the insulin sequences from different organisms from UniProt database. Let's begin by accessing the UniProt website. So I hope you know the web address for reaching UniProt. It is uniprot.org. Once there, we will search for the insulin protein sequences in various organisms. Start by entering the scientific name of each organism in the search bar. So here are the organisms we will be working with: human, pig, bovine, cat, degu, monkey, chicken, and guinea pig. For each organism, I have given the scientific name in the description. You can use that for doing the search. Say for example, for human, the scientific name is Homo sapien. For bovine, the scientific name is Bos taurus. For each organism, we have to locate the insulin protein entry and we have to download the protein sequence in FASTA format. Then we have to save all the sequences in a single file and we have to save it in FASTA format. So let me give demonstration for this, how to download the sequences from UniProt, how to save it in a FASTA file format. So we are in UniProt database. So first we will search for the organism one by one. First we will do the search for human. So go to the advanced option. So here we have to change this to protein name and change this to organism. The protein name is, we are searching for insulin sequence. The organism is, first we will search for human. The scientific name is a Homo sapien. So here you can see the Homo sapien. Select this and do the search. So the first entry P01308 is the insulin sequence in Homo sapien. So it is having 110 amino acid length. So click on this entry and go to this sequence and isoform section. So where we can see the sequence of human insulin and click on this download option. So we can see the sequence downloaded in FASTA format. So like this, we have to do the search for all other organisms. So we'll do the search quickly for the second organism. Go to advanced. So change this gene to protein name. Change this taxonomy to organism. So we are searching for insulin sequence. The second organism is pig. So whose scientific name is Sus scrofa. So do the search. So the first entry is the insulin sequence of pig P01315. Click on this, go to the sequence section and click on this download option. We got the sequence in FASTA format. Like this, quickly we'll do for the third one. The third one is bovine. So we'll change this to protein name and this to organism. So we are searching for insulin. The organism is bovine. Scientific name is Bos taurus. Click this and do the search. So the first entry is the insulin sequence of bovine. Click this, go to the sequence section and click on this download to download the sequence in FASTA format. So now go to the UniProt page, go to advanced, change this to protein name and change this to organism. So we do the search for insulin. So this time we do the search for cat. So this is for cat. Click on search. The first entry is the insulin sequence of cat. Click on the sequence option. Click this download to download it in FASTA format. The next is we do for Degu. Go to advanced, change this to protein name, change this to organism. So the protein name is again insulin. This time the organism is a Degu. The scientific name is Octodon. Octodon degus is the scientific name. Do the search and click the first entry and go to the sequence section and click on this download option. So we got the sequence in FASTA format. So now we do the search for monkey. So change this to protein and change this to organism. So the protein we search for again insulin. The organism is monkey. The scientific name is maca. So click on this and do the search. And the second entry is the insulin sequence of monkey. So click on this sequence and click on this download. So we got the sequence in FASTA format. Go to advanced. We'll do the search for chicken. Protein name, organism, insulin is the protein name. So chicken, gallus. Click on this. Click the first entry and go to the sequence section and click on this download option. So we downloaded the sequence in FASTA format. So one last search we will do for guinea pig. So change this to protein name and this to organism. And here the protein name is insulin and the organism is guinea pig. The scientific name is cavia. So for guinea pig, click it. So something wrong. Change this to protein name is insulin. Organism is, we had to search for guinea pig, cavia porcellus, this one. Click on the search button. So click on the first. So click the sequence and click download. So we got the insulin sequence in FASTA format for all the organism of our interest. So next what we have to do is that we have to save all the sequence in one single file and we have to save that file as FASTA file. So let me show how to do that. So open Notepad. So once if you open Notepad, so we have to copy the sequence one by one. So this is the first sequence. Copy this and paste it here. So then press enter. Go to the second tab. Copy the sequence. Ensure that you are copying everything, right? So along with the greater than symbol and paste it here. So this way we have to paste all the sequences we have downloaded This is the third sequence we have downloaded. Copy this and paste it here. So we'll do for the fourth sequence. Copy this and paste it here. So we'll do this for the fifth sequence. Copy this and paste it here. We'll do for the sixth sequence. Copy this and paste it here. We'll do for the seventh sequence. Copy this and paste it here. We'll do for the last sequence. Copy this and paste it here. Now we have pasted all the sequences in one single notepad file. So I am zooming this slightly so you can see it better. We know that all the 8 sequences are insulin sequence. So the first line starts with greater than symbol. So it is a FASTA file format. So there is an annotation in the first line. So no need to keep the entire thing. We will make it simple so that we can easily understand and we can interpret the results from multiple sequence alignment easily. We'll keep this as pig. We'll keep this as bovine, because we know that all the sequences are insulin sequence. So we'll make it simple. We'll make the header line simple. So this is cat. So I'm keeping this cat as. So this is from cat. So I rename this as cat. So the next sequence is a degu. So I rename this as degu. The next is monkey. So I rename this as monkey. So the next one is chicken. The next one is chicken. So rename it as chicken. The next one is Homo sapien. So we will rename it as human. The last one is guinea pig. So we rename it as guinea pig. Later you can understand why we do this, because while analyzing the results from multiple sequence alignment, it will be easy for you if you keep the header line simple like this. So we are going to save this file with all the sequences. So I keep the Homo sapien first. It is not necessary, but that's the order in which we have downloaded the sequences. So I am keeping this the first place, right? So we'll save this file as FASTA file format. Go to file, save as. So I'm saving this file in maybe desktop. I have a folder for applied bioinformatics, multiple sequence alignment. I am going to save in this folder. So the file name I am going to give as insulin dot FASTA. So everything should be within double quotes, because we are saving this file in FASTA file format. So here in this save as type, change this to all files. and save. So now we have saved this file in FASTA format, which contains the insulin sequences of all the 8 organisms. So I'm going to close this. So now we will do the multiple sequence alignment using Clustal Omega for the downloaded sequences. Clustal Omega is known for its accuracy and scalability, making it suitable for aligning large numbers of sequences quickly. It uses a guide tree and a hidden Markov model profile to generate accurate alignments. So now we will perform multiple sequence alignment for the downloaded sequences using Clustal Omega. So open the Clustal Omega website www.ebi.ac.uk/tools/MSA. So scroll down. Here you can see the different tools which are available for multiple sequence alignment in EBI website. So we go for Clustal Omega. So this is the online server for performing multiple sequence alignment using Clustal Omega. The first thing is that it is asking for sequence type. We have to choose what type of sequence we have. So our sequence is a protein sequence of insulin. So we have to choose this protein. Either we can paste the sequence here, or we can choose the file by using this choose file option. So since we have all the sequences in one single file, so I am going for this option. So I am going to upload this insulin.fasta file here. So it is asking what should be the output file format. We'll go with the default Clustal W character count. There are different other options available. We are not going to make any change here, and we are going to run this job by clicking this submit option. More options available, but we are not going to change any parameter. We go with the default parameter, and we will do the submission by clicking this submit button. So we need to wait for a few minutes for getting the results. So we got the result now. Click on this view result. So this is the result page of Clustal Omega. So the result is given in different tabs. So there are different tabs which are here. We'll explore one by one. So once the alignment is complete, we have to review the aligned sequence. So if you scroll down, so this is the alignment made by Clustal Omega for all the 8 sequences, insulin sequences of 8 different organisms. So here if you click this, this will show the alignment with color. So it will be easy for you to visualize and interpret. In this alignment result, you will notice symbols such as asterisk symbol, colon and dot. So we have to read the multiple sequence alignment column wise, not row wise. So we have to read like first column, second column, third column and so on. So each column there's a kind of indication. So first column you have asterisk symbol. If you have asterisk symbol on any of this column, a star symbol which indicates that the residues in that column are identical across all sequences in the alignment. This shows complete conservation at that position. The first position in this alignment is conserved. That is what this star indicates. So we have another symbol, colon. A colon indicates that the residues in this column are similar across all sequences in the alignment. Similarity means that the residues are not identical but share similar properties, such as being in the same biochemical group. Say for example, in this column, all the sequences does not have the same amino acid, but they have same physiochemical properties. So that is why here it is highlighted as colon. So the third symbol is the dot. So here you can see dot in this position. So what it indicates, A dot indicates that the residues in that column are somewhat similar. The similarity is lower than that indicated by colon, but still more than random. There is a kind of similarity, but not like the colon. So the use of these symbols help to quickly visualize and interpret the degree of conservation and similarity across the aligned sequences. So Clustal Omega also generates a guide tree and a phylogenetic tree. So this will help interpret the evolutionary relationships between the aligned sequences. So here you can see the alignment. So here the alignment is given based on some color code. So if you want, you can change the color code. By default, it is Clustal 2 color code. So the third option, as I said, is the guide tree here. So I can slightly zoom this tree. So hope this is visible now. So this guide tree shows the order in which sequences were aligned. So this will help in understanding the hierarchical structure of the alignment. Say for example, these two sequences were first aligned, then the pig sequence was taken and aligned with this human monkey, then this bovine sequence was taken and aligned with this 3 and so on. So this indicates the order in which the alignment was made. So there is another option here, phylogenetic tree. This also I can zoom slightly. I hope this is slightly better now. So this phylogenetic tree provides a visual representation of the evolutionary relationship between the sequences. So how to interpret the tree? So we should first look for cluster of sequences. So here we have a cluster of sequence, here we have cluster of sequence, here we have cluster of sequence. So we should look for cluster of sequence that are closely related. So these are closely related, these are closely related, these are closely related. These clusters indicate that the sequences share a more recent common ancestor. These two sequences share a more common ancestor here. These two share more common recent common ancestor here. These two shares recent common ancestor here. So we should also look for distantly related sequence. So we had to identify sequence that are more distantly related. Normally these sequences are placed on longer branches, indicating they have a more distant common ancestor. Say for example, this is a distantly related sequence. The sequence of chicken is a distantly related sequence. Then we should look for the evolutionary pathways. The branches in this tree, so these are the branches in this tree that represents evolutionary pathways. The length of the branches indicate the evolutionary distance between the sequences. And also we should look for the nodes and common ancestors. So these are the nodes. These points are the node points. So the nodes where branches split represent common ancestors. Sequences that split from same node are closely related. So here this split from the same node. So these are closely related. These two split from this node. So these are closely related. These two split from this and these are closely related. So we can use the tree to understand how the insulin sequences from different species are related. This can help hypothesize about their evolutionary history and how certain functional regions have been conserved or changed over time. So now we move on to MUSCLE. MUSCLE stands for multiple sequence comparison by log expectation. Is another powerful alignment tool known for its high speed and accuracy. It employs iterative refinement techniques to improve the quality of alignments, making it particularly effective for data sets with moderate number of sequences. So now we will open muscle and we will import the same set of insulin sequences from 8 different organisms. So we will first reach the muscle. So again go to the same website www.ebi.aci tools MSA. So go to this website and scroll down where we have the option for muscle. Click on that. So this is the online server muscle for performing multiple sequence alignment. So I can reduce the size. I zoom out this slightly for better view. So here we have to paste the sequence either, or we can upload the sequence. We will upload the sequence from here. So again we are using the same file, FASTA file, insulin.fasta, which contains the insulin sequence from 8 different organisms. The output format, we are using the same default one without changing any parameter. Default parameter, we submit the job. Now the job is running. We'll wait to get the results. So we got the results. So again, here also the results are organized in the same way. So we have the tool output. So in the bottom we have alignment with color. Click on that. So whatever I explained for asterisk symbol, colon and dot, that is applicable for muscle also. So here also we have the alignments. So it's using the color code Clustal 2. If you want, you can change the color code. So we have the phylogenetic tree. Same explanation what I gave for Clustal W, that is applicable for this. We have results viewer. We're not going to use anything here. So we have a result file. We can download the results. We have the submission detail. So here this is the place where you can infer most of the things. Okay, so which residues are highly conserved, that we can figure it out from this output. You can get some idea about the evolutionary relationship from this phylogenetic tree. So now we got the results from both omega and muscle. So what you have to do is that you have to compare the results which you got from Clustal Omega and muscle. So you have to note any differences in the conserved regions and variations between the output from Clustal Omega and muscle, and you have to discuss how the choice of alignment tool might impact the analysis. Now we will analyze conservation of functional regions in insulin. Insulin has several important functional regions, including the A and B chains and the disulfide bonds that stabilizes its structure. So just go through the alignment result and find out, is there any cysteine residue that is highly conserved in the insulin sequence of all the organism that may be important for the formation of the disulfide bond. The single letter code of cysteine is C. So in this alignment, we will look for, is there any cysteine that is highly conserved. Here you can see there is a cysteine that is highly conserved in this position. So here also you can see there is another cysteine that is conserved in all the 8 organisms. There is another cysteine that is conserved all the 8 organisms. So why it is conserved? Even during the evolutionary process, the cysteine at that position is not changed because it is very important for maintaining the structure. To maintain the integrity of the structure of insulin, the cysteine at these positions are important. That what we can infer from the multiple sequence alignment of the insulin sequences. Now let's move on to some self-evaluation task. So based on the learning from the multiple sequence alignment exercise, perform a self-evaluation by conducting an in-depth analysis of the alignment results using a different set of sequences. So you have to retrieve the cytochrome C sequence in FASTA format for the following organisms from UniProt: human, mouse, rabbit, horse, cow, chicken, frog, zebrafish, and fruit fly. So you have to perform multiple sequence alignment using Clustal Omega and MUSCLE, and you have to compare the alignments and analyze the results. By completing this exercise, you gain hands-on experience in performing multiple sequence alignments using Clustal Omega and MUSCLE. You learn how to download protein sequences from UniProt and analyze the aligned sequences to understand evolutionary relationships and sequence conservation. This knowledge is crucial for conducting bioinformatics research and analyzing protein sequences in various biological contexts. The self-evaluation task further solidified your understanding and ability to critically assess multiple sequence alignments. Thank you for watching. Don't forget to like, subscribe, and leave a comment if you have any questions. Stay tuned for more tutorials.